Okay, so let's get started. Hello everyone. Welcome to our today's Museum seminar. It's absolutely my pleasure to welcome our today's speaker, Dr. Ludger Klein, who is from Munich. So I met Ludger last year on Cold Spring Harbor meeting on the immune tolerance meetings, and then he really gave a marvelous seminar, which I'm so blown away. So I decided to invite him back on our museum seminar to introduce his work to more broad audience. So a very brief introduction about Ludger. So Ludger actually completed his undergrad study in biology in University of Cologne in Germany, and then continue with his graduate study in Heidelberg and Cologne. So after he completed his PhD training in genetics in Cologne, he decided to pursue postdoc training. At the beginning was in DKFZ in Heidelberg, and then he moved to U.S. in Boston. Did his later part of his postdoc training in Dana-Farber Harvard Medical School in Boston. So from early 2000, Ludger actually started his independent research program as a junior group leader in Research Institute for Molecular Pathology in Vienna, in Austria. And then he moved to back to Germany, all the way up to from associate professor all the way up to the full professor in Munich in Ludwig Max Millions University in Germany. So a general overview of Ludger's lab is he's mainly interested in studying immune tolerance, basically mainly focusing the thymus. As we know that tolerance to self is a fundamental property of the immune system, which breaks down can lead to autoimmune disease such as multiple sclerosis and diabetes. So selection process in the thymus are really essential for the generation of useful and self-tolerant T cell repertoire. By using genetic manipulated mouse models, his lab mainly aimed to understand how cell biological and the molecular properties of thymic microenvironment, how they orchestrate T cell fate decision during thymic selection. So several different aspects in his lab regarding to the immune tolerance, including B cell and then central T cell tolerance, negative selection versus induction of regulatory T cells, and then also his lab interested in studying how the promiscuous expression of self-antigen in the thymus. And last but not least, his lab also studied the generation of MHC peptide ligands for T cell repertoire selection. So I think Ludger gonna to introduce us a lot of his recent work regarding to the thymic specific T cell repertoire, and then thank you so much for sharing your study with us today, and then we are very much looking forward to it.
Yeah, so you can still hear me all right? Yeah.
Yeah, it's perfect.
Yeah. Thanks, John, for the kind introduction and for having me today. So it's a pleasure to share our work and as John alluded to, so our lab works a lot on central T cell tolerance, but what I thought might be fun to discuss with you today is the other side of the coin, so to speak. That's positive selection of the T cell repertoire, and this is a talk I haven't given too often yet. Many of the slides have just been generated, so if I get confused by my own slides, be kind to me. I apologize in advance. So most important slide first. So essentially, I mean, 99% of what I'll be talking today is the work of a fantastic PhD student and then postdoc in the lab, Elisabetta. She has left the lab in the meantime. Now she's traveling, and I hope that she will pursue an academic career. So Elisabetta um, has been in charge of most of the work. And what I will be talking about is a thymus-specific protease called cathepsin L and its role in CD4 T cell positive selection. So this is a slide I like to use initially to introduce a topic. So I don't want to insult your your expertise. So this is the most simplistic scheme. So if you want to tell your mother what the thymus does, you can look at it as a T cell school, right? And you have these hopeful progenitors from the bone marrow. They enter this kindergarten or school. And then there is two critical aspects. And if you take a close look, you see they grow arms here, right? And so they can sense the world, right? And this is the T cell receptors. You know, these are being rearranged when the cells have entered, the progenitors have entered the thymus. And then, of course, these T cell receptors, they have to be tested for two properties, and that's depicted here. So in simplistic terms, you could say a T cell repertoire on the one hand needs to be useful and on the other hand needs to be harmless, right? And you all know what these two terms mean. Namely, on the one hand, you have to be self-MHC restricted, so that would be useful, and harmless, of course, the repertoire has to be selected against the dangerous ones that could attack the body's own tissues, right? So that would be tolerance. And what I want to do today is I will use a quite extensive introduction, right, to lead up to the topic, because I don't really know, I mean, who I am talking to today. So so not to lose anyone who has just started on the topic, I will use a rather extensive introduction and then maybe be a bit shorter on the actual data in the end. So this takes us back many, many years. So in the 1980s, 1990s of the last century, I mean, it became clear that, I mean, these processes of generating a useful and harmless repertoire, that both processes essentially involve interaction of the T cell receptor with self-peptide MHC complexes in the thymus. And at the time, it was called the selection paradox, right? So you have two diametrically different outcomes. On the one hand, survival, right, and lineage commitment. And on the other hand, removal, apoptotic death of a cell if it sees something with its T cell receptor. And that, of course, is kind of a paradox, if you wish. And so here, as you can see, so this is very early work from Pippa Marak and John Kapler, and they discussed, I mean, the underlying principles of this paradox, and they were suggesting an explanation for this paradox, which at the time was called the altered ligand. Or here's the paradox, right? So here you see a peptide MHC complex on a positively selecting cell. At the time, it was clear that this was thymic epithelium. It was not specifically clear that these are cortical thymic epithelial cells, as we know now. And then there was a second compartment, which was believed to induce tolerance. And here they called it thymic macrophage. Nowadays, we know that this is a complex mixture of cells that can be tolerogenic, and that's dendritic cells, medarothymic epithelial cells, and thymic B cells. Anyhow, regardless of that, I mean, so the paradox is, if you have a peptide which is visible on both types of cells, you use that peptide for positive selection. And then the cell sees the same peptide again in a compartment which is tolerogenic. Shouldn't you end up with a repertoire which is largely eliminated as a consequence of this dual encounter of the same ligand for both positive and negative selection? And the scenario they were suggesting at the time, the hypothetical scenario, to resolve this paradox. Hoppla, now I'm going the wrong way. So they called it the altered ligand hypothesis. And so what does that mean? So the hypothesis they were putting forward was that if these positively selecting cells make a deliberate effort to present peptides that are different from the ones that are seen on the negatively or tolerance inducing compartment, you should be fine, right? Because then the two processes don't cancel each other out. So obviously then, I mean, how would you test that hypothesis? And Pippa and John actually moved on in the following years to address that, because the obvious test of this hypothesis was, if you were to know those peptides which are seen on those cells as opposed to the peptides which are expressed on those cells, is there a non-overlap? Are they distinct or not? So what they did, and this experiment is shown here. So now we're in 1992. So this is a heroic experiment at the time where they eluted MHC class 2 bound peptides from enriched cortical epithelial cells of the thymus, and compared those to not thymic dendritic cells and B cells, but splenic antigen presenting cells, splenic MHC class 2 positive cells. And without going in too much detail, so here, I mean, so they identified a handful of peptides, and here 6 are shown, and this is the abundance of the respective peptide on this cell type, so on the positively selecting cell type, and on other cell types that are believed to be representative of the tolerance-inducing cell types. And as you can see, it's not binary. It's not 0 and something here and vice versa. What you see is these peptides are shared. There is quantitative differences, supposedly, but by and large, I mean, whatever you find on the positively selecting cells, there is a high likelihood that you will also find it in the spleen and vice versa. So on the basis of that observation, and so just as a fun fact here, so this is not a trivial experiment. So if you look into the material methods, so this is spleens and thymuses isolated from 100 to 375 mice for each class 2 preparation. So just to give you a flavor of the technical challenge that these experiments present. On the basis of those findings, however, then, I mean, they put their own hypothesis to rest, right? Because obviously it's not that all the ligands that are on cortical, on the positively selecting cortical epithelial cells, would not be found on the tolerance-inducing compartment. And you all know, I mean, so in the years after, I mean, what has until today has become the prevailing model of how positive and negative selection, or how this selection paradox can be resolved, is this affinity-avidity model, where it's less whatever a compartmentalization, but a gradual, a sliding scale of affinity-avidities of the T cell receptor and its peptide MHC ligands that makes the difference between positive and negative selection. So that is there is a lot of evidence in favor of that model. But of course, this model, there is a couple of features of the system that this model does not incorporate. So is the selection paradox solved? So here's our affinity model. And then there is a couple of facts that are not incorporated into the model. So you could ask, I mean, why then, if it works like this, why then did evolution come up with a scenario where positive and negative selection are largely separated, not only spatially, but also temporally? So once again, to remind those of you not looking at the thymus on a daily basis, so positive selection occurs in a compartment which is called the cortex. And again, this is the cell type that presents self-peptides for positive selection, the cortical thymic epithelial cell, and then subsequently the positively selected cells move into the medulla, where they interact with those tolerogenic antigen-presenting cells that would be medullothymic epithelial cells and dendritic cells. And all this, of course, is not reflected in this affinity-avidity model. Why would you have a need, obviously, for a dedicated antigen-presenting cell for positive selection? Again, once again, so the only cell that can positively select magically, miraculously, seems to be the cortical thymic epithelial cell. If those don't have MHC molecules, you will not positively select any cell. And last not least, I mean, these cells, why do these cells, and I would call it, try to be different? And what does that mean? So cortical thymic epithelial cells, they seem to use, or it's clear that they use proteolytic pathways for the generation of both MHC class 1 as well as MHC class 2 bound peptides that are different from those that other antigen-presenting cells use. So that just schematically, so if this is a cortical thymic epithelial cell, if this is anything else, so for instance, a dendritic cell or a B cell or a medullary-thymic epithelial cell, I suppose you are aware of the thymoproteasome, so beta 5T, which on which Yosuke Takahama has worked for many years and has published fantastic work. So this seems to be a proteasomal subunit which is only expressed in cortical thymic epithelial cells, whereas other cells use the housekeeping proteasome or the immunoproteasome to generate class 1 bound peptides. And then there seems to be a mirror image, a correlate of that in the class 2 pathway, and that is a differential expression of cathepsin L only in cortical thymic epithelial cells, whereas cathepsin S, so another lysosomal proteases, seems to have a analogous function in other APCs. And then by inference, you would say probably this points towards the presentation of what I would like, or what people call private peptides on cortical thymic epithelial cells, right? Which might be different from the public peptides. And then again, then it would be easy. Okay, so do the Kaplan-Meier experiment, right? Show me the peptides that are on cortical thymic epithelial cells versus other APCs. And again, I mean, so now we are roughly 30 years later, and we still don't have a comprehensive idea of which peptides are presented. And so the next slide is just to, the problem or the issue is a little bit related to dark matter. I mean, if you think about astrophysics, right? I mean, there is good evidence that dark matter must be there, but we don't know what it is, right? And astrophysicians measure effects of astrophysicists, not physicists. Astrophysicists measure effects of dark matter and infer that dark matter must exist. But obviously, as the name says, no one has ever seen it, right? And I think the analogy to the thymic peptidome is obvious, right? So we have to infer from experimental observations to the nature of these peptides that mediate positive selection. So here's an experiment that has addressed, and again, so this is 20 years back. So Chiche in Sasha Rudensky's lab. I mean, is there evidence that cathepsin L and cathepsin S, so these two differentially expressed proteases, that they actually generate different peptides from substrates, right? And if you look at this, so this is fibroblasts. So this is not the real thing. This is not cortical thymic epithelials, but this is fibroblasts transduced with cathepsin L or cathepsin S, and then you elute a couple of abundant peptides. And you ask, I mean, are they differentially present? And obviously they are not, right? So if you look at this white bar and this black bar, many of the peptides seem to be the same. Yeah? So again, that would say it's not black and white, but what I would like to draw your attention to here is peptides. So here, for instance, that is a peptide that only shows up when substrates, and again, it's unknown what these substrates are, but this peptide only shows up when cathepsin S generates class 2 bound peptides, and this vice versa, this thing here only seems to show up when cathepsin L processes peptides for class 2 bound peptides. So all this, so we are coming back to that scheme here. So this, of course, is an extreme view and again, I mean, the Kaplan-Meier experiment has shown that this extreme view is not the case, but what more likely might be the case is that on these cortical epithelial cells, you have two principally different cohorts of peptides, namely shared peptides, public peptides, but on top of that, private peptides, right? So it's probably conceptually more appropriate to think in that kind of a model rather than an either-or model, right? Then, of course, the question is, how important is that? What happens if you take away cathepsin L, right? And once again, so here's our dark matter. I mean, no one has measured whether or not peptides disappear when you eliminate cathepsin L, but the experiment as such has been done before. And here's the outcome. So this is work again from Sasha Rudensky's lab more than 25 years ago. This is the thymus of a cathepsin L knockout mouse. And so this you stain for CD4 versus CD8. So you all know this picture. And I hope what you can appreciate is that there is a dramatic reduction in CD4 T cells, right? So you take away cathepsin L, a dramatic reduction. And again, consistent with the idea that peptides disappear, which are necessary to select a reasonable fraction or in any other way support positive selection of CD4 T cells. So this is kind of the state of the art, and has been the state of the art for many years. That has never been really followed up, and that is what Elisabetta decided when she entered the lab to decide to take a closer look at. And in essence, the question is, one question is, what are these, right? So you get a reduced CD4 T cell compartment. So the question is that just less of the same, right? So if you imagine you have millions of T cell receptor specificities here, probably every T cell receptor might occur 2 or 3 or 4 or 10 times, right? And do I just condense the entire repertoire but retain everything, so to speak? Or is there a selective loss of particular T cell receptors because particular peptides might be missing, while other T cell receptors clones are retained in the repertoire? And then, of course, the question, I mean, these retained cells, obviously, these are T cell receptors, cells that can be selected in the absence of cathepsin L. Are these functionally normal, right? So that's what Elisabetta set out to address. So first of all, the straight cathepsin L knockout mouse, and that is kind of a dirty little secret of the system, is so if you use the straight knockout mouse, so cathepsin L is not so cortical epithelial cell specific as, for instance, beta 5T is in the class 1 world, right? So and so cathepsin L, for instance, is also expressed in the skin, not in antigen presenting cells of the skin, but obviously it does something there. And this mouse has a dramatic skin defect, so there's alopecia over time. So these mice lose their fur, etc., etc. So they look like a nude mouse, but they are not, right? So Elisabetta decided to maybe we can use a cleaner system, which would be a conditional knockout of cathepsin L only in thymic epithelium. So for those not thinking about that thymus on a daily basis, we don't have a conditional Cree driver way we could selectively knock out cathepsin L only in cortical epithelial cells. So when we use this is a FOXN1 Cree transgenic, right, combined with a conditional cathepsin L allele. So this will knock out cathepsin L both in cortical thymic epithelial cells and medarothymic epithelial cells. Now, considering that medarothymic epithelial cells barely express cathepsin L because they express cathepsin S, and vice versa, the cortical thymic epithelial cells, they express cathepsin L but not S. I think it's kind of a proxy to a conditional knockout of cathepsin L in cortical thymic epithelial cells. So first important observation is this conditional knockout that absolutely recapitulates the previously described phenotype of the full knockout. So you see this dramatic reduction in the CD4 single positive compartment. So once again, so why is that now? The most simple explanation, if you think about this scenario here, that would be that you have complete absence of peptides or class 2 on cortical thymic epithelial cells because this protease is absent. But that is clearly not the case. So if you look at these, at the cortical thymic epithelial cells in the cathepsin L knockout delta tech versus the wild type mouse, you see that total class 2 levels are absolutely superimposable, right? So that cannot be the explanation for a reduced CD4 T cell compartment. So, and again, consistent with the idea that these remaining class 2 molecules on a cathepsin L delta tech cortical epithelial cell, they present something, but which by definition cannot be dependent on cathepsin L to be generated, but would be what we would like to call public peptides. What I haven't discussed with you yet is cathepsin L, so that's a complication of the system. Cathepsin L has not only been implicated in the generation of MHC class 2 bound peptides, but also in the processing of the invariant chain, right? So I guess you know the invariant chain as kind of a placeholder that occupies the class 2 peptide binding groove on nascent MHC class 2 molecules, and that then has to be cleaved by proteases, including cathepsin L, but obviously in a redundant manner by other proteases, to ultimately release the invariant chain or the clip fragment of the invariant chain. So one possibility is now, if you now knock out cathepsin L, could it be that you end up with an MHC class 2 peptide ligandome which only consists of clip molecules, right? And again, if you only have one species of MHC peptide ligands, that could be an explanation why your class 2 CD4 T cell compartment collapses. So there is a beautiful antibody which allows us to stain this complex of MHC class 2 and clip on the surface. And what you indeed see is, once again, so blue is a cortical thymic, or is cortical thymic epithelial cells of a wild type mouse. You see that we have an increase in MHC class 2 clip expression on cortical epithelial cells in these delta tech mice. This is, and you know that is very difficult to quantify. What does such a shift mean? So do we have evidence? So, but luckily there is another antibody, and this is an antibody that recognizes, so to speak, anything which is not occupied by clip. Right? So let's call it MHC class 2 non-clip. And as you can see here, reciprocally, non-clip, so occupation of class 2 on these delta tech CTEX is reduced, right? But it's compared to the MHC, to the control staining of an MHC class 2 knockout cortical thymic epithelial cell, there still seems to be a huge amount or huge fraction of class 2, which is occupied by something, again, whose identity we don't know, right? But it does not seem to be all clip. And finally, so here is a particular ligand, and this is a fantastic antibody that recognizes class 2 complex to one particular peptide. Again, this is a very frequent, abundant peptide. And as you can appreciate, this is reduced, but it's by far not gone, right? So by inference, this cannot be, so a deletion of cathepsin L does not lead to a clip-only peptide MHC ligandome on cortical epithelial cells, but it's rather, all these findings are rather consistent with many public peptides being retained, and yet you get this strong reduction in CD4 T cells. So once again, so this is what I've shown you so far, right? So this is a wild type situation. You select in the presence of cathepsin L in cortical thymic epithelial cells, and you get a repertoire of a given size. So that's our wild type size. And if you eliminate cathepsin L, you get this contraction. So what you need to consider, or looking at that, what is important is that there is two explanations for a smaller CD4 single positive compartment. And one would be a true bottleneck in positive selection, right? So just numerically, you generate less CD4 single positive cells. And an alternative scenario would be, maybe in principle, I generate the same number of single positive cells, but these are different CD4 single positive cells, which are hyper-susceptible to negative selection by these other antigen-presenting cells, right? So and in the end, the net effect would look pretty much similar if you would just look at X% of CD4 single positive cells. So how can you distinguish these two scenarios? So first of all, let's look at this scenario. If this here were the case, a prediction would be, if I eliminate the negatively selecting influence of these type of cells, of the hematopoietic antigen-presenting cells, I should rescue a CD4 single positive compartment of substantial size. And this is depicted here. So in principle, and again, this scenario of increased negative selection, I hope you can appreciate that this is a bit reminiscent of, it's not synonymous, but it's quite reminiscent to this model, or to the paradox that John and Pippa back in the days already suggested, right? So that you would re-encounter the same peptides. So you are positively selected on something which you again see on negatively selecting cells. So here's the scenario once again. So, and if that were the case, if you take away negative selection by those cells here, do you rescue the CD4 single positive compartment? So how do you do that? So we do that in bone marrow chimeras. So this is the experiment. So this is the wild type setting. So this is you put wild type bone marrow into a wild type mouse, so to speak. Yeah, let's call it cathepsin plus, of course, right? Genetically. So this is our reference population, right? So here is an experiment that has been done by others and ourselves many, many times before. So if you put MHC class II deficient bone marrow into a wild type mouse, so you indeed eliminate a certain fraction of negative selection, and that manifests in an increased size of the CD4 single positive compartment. That's what we are looking at here, right? So this delta here is believed to represent the impact of hematopoietic antigen presenting cells on negative selection on MHC class II restricted cells. So this is kind of the wild type situation. Now, if we do the same exercise in a cathepsin L knockout, and we can also do that in a cathepsin L delta tech mouse, you see that nothing whatsoever happens, right? So to make a long story short, so the message of that observation is here, you see there is no delta here, right? So regardless of whether you can negatively select by hematopoietic antigen presenting cells or not, as in this case, you don't rescue the CD4 single positive compartment. So that is not a question, I suppose, right? That's a discussion in the background. So I would say on that basis, we can discard this possibility, and this rather points to a bona fide defect in positive selection. So how can you test that more specifically? So one way of testing that is, and that is what Elisabetta has done with in essence, every class II restricted T cell receptor transgene we could get a hold of. We generated bone marrow chimeras. So this is, so for instance, what you see here, this is one particular T cell receptor. In essence, it doesn't even matter what the specificity of that T cell receptor is. So as you would expect for a transgenic class II restricted T cell receptor on a RAC knockout background, it nicely generates a bias towards CD4 single positive compartment. Now we have an exaggerated, highly enlarged CD4 T cell compartment, because every cell comes into the system with a monoclonal T cell receptor that is MHC class II restricted. So every cell will go this way and not that way here. So this is blank, so to speak. They all go here. If you put this, if you send the same cells through a system which does not express cathepsin L in cortical thymic epithelial cells, it's very dramatic. So this is absolutely blank. So this is not even quantitative, as in a polyclonal repertoire. There is, in essence, there is not a single cell that makes it through the system, right? And this is one representative example with the LLO 118 T cell receptor. We can do the same exercise with the OT2, which you are maybe more familiar with, and ovalbumin specific. So these are all antiforeign specific T cell receptors. None of those are selected, right? And in a black and white fashion, essentially. So which tells us there is a true bottleneck in positive selection, because these monoclonal cohorts don't make it at all. which then again raises the issue of, or the question is, what is the residual repertoire in cathepsin L-delta tech mice composed of? So to address that question, we use a trick that has been used by others before, and that is shown here. So I guess you know or can appreciate that comparing repertoires between individual mice simply on the basis of the non-stereotypicality of repertoires from mouse to mouse to mouse. I mean, you cannot do that in a fully alpha-beta polyclonal repertoire. So the trick we are using here is we use a mouse with a fixed T cell receptor beta chain, so depicted in blue here, right? And of course, I mean, these T cells that arise in such a mouse, they use endogenously rearranged alpha chains paired with a fixed, always with a constant beta chain, right? Which then has the advantage that you don't have to do single cell sequencing of the T cell receptor, but you can do bulk RNA-seq of the T cell receptor alpha chains and generate a nice inventory of clonotypes that are used in this animal. So here is the experiment. So this is how these fixed beta cathepsin wild type, so cathepsin plus mouse looks like, right? So you have like 9%, looks pretty normal with regards to the size of the CD4 single positive compartment. And it nicely recapitulates the reduction, right? When there is no cathepsin L in cortical thymic epithelial cells. And so what we are doing now, or what we are looking at here is now, so we sequence T cell receptor alpha chains of those and sequence T cell receptor alpha chains of those. And what you see here is, so this is the average frequency of a given clonotype in a cathepsin L sufficient mouse, plotted against the average frequency of that particular clonotype in a cathepsin L delta tech mouse. And if there was no difference, what you would see is that there is a diagonal. Every T cell receptor would be equally frequent in either genotype. But what the actual outcome is that there is indeed some T cell receptors which are shared. So by definition, they are selected also in the absence of cathepsin L. But there is a lot of T cell receptors here depicted in blue, which don't show up at all in the repertoire of the cathepsin L delta tech mouse. So we are looking at a total of around 10,000 T cell receptors, and 4,000 actually seem to disappear. And interestingly, it's not only that T cell receptors disappear, but when you don't have cathepsin L, there is also so-called newcomers. So there is T cell receptors which only show up in the repertoire when you don't have cathepsin L. And again, so this clearly shows that there is high degree of selectivity in the loss of T cell receptors. So in other words, it's not less of the same, but you seem to lose some T cell receptors while you retain. So these are the lost T cell receptors, and these are the retained T cell receptors. Apparently, cathepsin L independent T cell receptors in their positive selection. So how does that reflect onto the repertoire composition? And the readiness, so to speak, of the repertoire to respond to pathogens. So here you see precursor frequency in the naive repertoire of a wild type mouse as opposed to a cathepsin L delta tech mouse of a number of T cell receptor specificities. So this, we are using tetramers, and we use the Mark Jenkins trick, right, of quantifying the total number of cells in the naive repertoire. I suppose many of you know the trick. So you pool all CD4 T cells you can get a hold of in the mouse, you label them with a tetramer, you do a max enrichment of the tetramer, and then you do a dump staining, etc., right? And then you can really quantify in an entire mouse immune system. For instance, in this case here, there is around 10 cells which recognize this particular antigen, right? And you see that this is dramatically reduced, and it's pretty much proportionally reduced to the loss of clonotypes in the repertoire. And the same is true for another foreign epitope, right? But then there was a curious observation. So here, so this is an MHC class 2 tetramer that is that detects a Listeria LLO specific T cells restricted to IFB. So that's the class 2 molecule of BLEC6. And you see the number is not affected, right? Although we have this dramatic reduction of CD4 T cells and we have this selective loss of clones, there seems to be a similar number of cells that are stainable with that tetramer. So then Elisabetta went on to ask, I mean, does that really translate into a similar efficacy of the two types of mice to respond to immunization with this particular antigen? And this is an experiment here. So once again, so this is what I just showed you. So this is the naive repertoire of tetramer positive cells. So we have about 400 cells in the entire repertoire, regardless of whether the mouse is wild type or cathepsin L delta tech. But now when we immunize this mouse, what you see is that as expected, so this is the max enrichment Jenkins protocol. This is direct staining, right, without max enrichment. So you just gate on CD4 T cells and you can easily see these cells with the tetramer staining because they are massively expanded, right? So that's what you would expect. However, despite a similar precursor frequency, there is a pretty wimpy expansion of those cells in the delta tech system, which raises the question of, are these cells that normally would nicely respond by expansion, are these clones actually gone? And is that compensated by the emergence of other clones? So does that explain this weak expansion? So how can you address that? So once again, we go, so this is a complicated slide, but bear with me. So what you see here is, once again, this time now we immunize a fixed beta mouse, wild type, cathepsin plus, with the LLO peptide. And then what we do is we sequence the T cell receptors on these expanded tetramer positive cells. And these are the top 10 T cell receptors. So here you see the clones, but doesn't matter in the end. So this, and then of course we can go back to our inventory, which we established. So this is the color code corresponds to these top 10 T cell receptors. So these are the best responders in a wild type setting. And then of course you can ask this T cell receptor. So the best responder here, is that a blue T cell receptor, if you wish, in our repertoire inventory, or is that a cathepsin L independent one? And curiously, you see that there is a pretty similar contribution of apparently cathepsin L independent T cell receptors and cathepsin L dependent T cell receptors to the immune response in a wild type mouse. So these cells should be around in the cathepsin L delta tech mouse, but why then do they not properly expand? So this suggests so that raises the possibility that the same clone, the same T cell receptor or CD4 T cell carrying the one and the same T cell receptor is functionally different when it's selected in the presence or absence of cathepsin L. Now, how do you test that? So what Elisabetta decided to do is, let's take this best responder, so to speak, in the wild type setting. And so we know it's T cell receptor alpha chain. Remember, this is paired with a fixed T cell receptor beta chain. So what we did once again is generate T cell receptor transgenic animals carrying this particular T cell receptor and now breed them onto a wild type background or to an cathepsin L deficient background and ask, first of all, is this T cell receptor selected as you would expect from inference from this picture here? And is it functionally normal? So here's the picture. So the T cell receptor now is, so this is this purple T cell receptor here. From now on, we will refer to this as LM54. And as you can see, in contrast to all the T cell receptors, which I showed you before, remember, the OT2, the AND is not selected at all when cathepsin L is absent in thymic epithelial cells. This T cell receptor is nicely selected, perhaps not as efficiently as it is in the wild type, but you get a substantial fraction. 30% of all your thymocytes are cells, CD4 single positive cells of that specificity. And again, this is for the aficionados among you. So this is maturation at the CD4 single positive stage. So they nicely undergo this post-positive selection maturation, indistinguishable from what you see in the wild type setting. And that now, of course, raises or gave us the possibility to ask, I mean, is this really, are these cells normally selected? So the first thing that Elisabetta did was So this is staining of NIR77. So this is a reporter of the T cell receptor signaling strengths. And so now we are looking at consecutive stages of T cell differentiation. So this is pre-positive selection, double positives. So these cells presumably have not received any T cell receptor signal yet, and indeed they are identical with regards to NIR77 staining intensity. But then the blue ones, the cells selected under wild-type conditions, they nicely upregulate NIR77 at these consecutive stages. And the first striking observation is these cells, although they upregulate CD69, which is another indication of them having received a positively selecting T cell receptor stimulus, this seems to have been weaker. Right, and this footprint, so to speak, of selection on a weaker signal seems to kind of persist through stages of selection downstream of the double positive of the selection event. So remember, these cells here are in the medulla. They are not in contact with cortical epithelial cells lacking cathepsin L any longer. So this is CD5. This is another reporter of the intensity. And you know CD5 is a rheostat of the signaling intensity. So the more intense your positively selecting signal is, the higher CD5 is believed to be expressed. And you can see here this normal trajectory of CD5 expression concomitant to progressive differentiation at the CD4 single positive stage. Again, this is consistent with the idea that these cells receive weaker signals. They are positively selected, but somehow this seems to happen in the context of weaker selecting signals. And curiously, now, if you look at, so CD5 is an inhibitor of T cell receptor signaling. And indeed, if you take those cells, so now we take the mature M2 cells, they are a couple of days downstream of positive selection. If you take those, put them in a culture dish and confront them with their agonistic cognate antigen, the LLO peptide, what you see is that the cathepsin L deficient or cathepsin delta tech selected cells, they are hyper responsive, consistent with lower CD5 expression, sensitizes your T cell receptor signaling chain. So you are indeed the IC50 of responsiveness. So as measured by CD69 upregulation as a function of peptide dose is lower for the LM54 cathepsin delta tech cells. So you are indeed hypersensitized. So then to get so they are different. They are not the same cell, curiously. And to get a more global view of changes in these two cell types, the selected, so apparently we are talking about a CD4 single positive cell carrying the very same T cell receptor, but selected in the absence of presence of cathepsin L. When you do a global transcriptomic comparison, so this is an MA plot. So this is the level of the average level of expression, and this is the differentiality, so to speak. Everything in red is significantly upregulated when you're selected in the absence of cathepsin L, and these things are selectively downregulated when you're selected in the absence of cathepsin L. So CD5 you have already seen at the protein level. And what struck us, I mean, when you do gene set enrichment analysis, it seems that these cells that have matured to the M2 stage, so this is transcriptomics of cells far downstream of positive selection, right? M2 CD4 single positive cells. So these are downregulated traits when you are selected in the absence of cathepsin L, and you see translation, initiation, translation, elongation, cytoplasmic and ribosomal proteins, ribosome, etc., MYC targets. So all this seems to look like a less biosynthetically somehow less poised state. And indeed, so you can, is there really less translational activity? So there is a very cool flow cytometric assay for translation using puromycin. So these are pre-positive selection cells. So this is the basal state of a cell. This is the CD69 double positive stage, and you see that the blue cells, the wild-type selected cells, they nicely upregulate or initiate or become more active with regards to translation. The orange, the cathepsin delta TEC selected cells also do that, but not to that extent. And once again, the striking thing is this footprint lingers on, right? So although this cell here might easily be 4 or 5 days downstream of the positively selecting signal in the absence of presence of cathepsin, and yet it still carries this footprint of the way in which it was selected. And finally, so if we take those cells, so now we take the very same clonally identical cells with regard to their T cell receptor, either selected in the presence or in the absence of cathepsin L, mix them at a 1-to-1 ratio and put them into a wild-type recipient and ask, are they homeostatically maintained in a similar manner? You see that the cathepsin L delta TEC selected cells, they have a massive competitive disadvantage, so you lose them over time in comparison to their sisters and brothers, which have been selected in the presence of cathepsin L. So I think this is my last data slide. So summary, cathepsin L is essential for normal positive selection. So bona fide, so the defect in cathepsin L deficiency is a bona fide defect in positive selection. It's not necessary to avoid excessive negative selection. Remember, in the context of this encounter of antigen scenario, we now know that we can quantify the extent of clonal loss in the repertoire. So around half of the CD4 T cell repertoire is essentially cathepsin L dependent. So these cells truly disappear. They are not selected at all if you don't have cathepsin L. But then on top of that, it turns out that among the cells that are retained in the repertoire, remember, so these are the gray, this is the gray diagonal in our repertoire analysis. We think that many of these cells, we did the same exercise for a second T cell receptor transient, and in essence, we get the very same results. So we think the fate of this LM54 T cell receptor is representative of a large fraction of T cell receptors that are apparently cathepsin L independent with regards to showing up in the repertoire at all. But we think that these cells are functionally impaired, as read out, for instance, responsiveness and homeostasis under competitive conditions. So once again, so this is kind of a visual, and again, I mean, all that against the background of the identity of those suspected private peptides being a huge black box, right? So, and this is up for the future, right? And with that, I'd like to thank you for your attention. So I mentioned Elisabetta. So again, almost single-handedly, although there is a large number of people on this slide, right? I mean, so as I said, I mean, 98% of the work done by Elisabetta. So these are collaborators in Hanover, and Stephen Daly in Australia, who helped us with repertoire analysis. The conditional knockout of cathepsin L is from Thomas Reinheckel, etc. And these are other collaborators. So thank you very much for your attention, and I'm happy to take questions.